Hello guys, good afternoon. So for today's video, I am going to uh, share my summary of learnings in Biology 3. But first, let me introduce myself. My name is Felyun Magpayo. I am a Biz Ed Science student, second year. So this is the last task in this semester, which is to have a video presentation about the summary of learning in this subject. So let's start. So there are three units that we need to have a summary. Uh, Unit 5, 6, and 7. So let's start with Unit 5. So Unit 5 entitled Microbial Metabolism. So let me define what is metabolism. Metabolism is the build-up and breakdown of nutrients within a cell. This chemical reaction provide energy and create substances that sustain life. Metabolism is an emergent property of life that arises from orderly interactions between molecules. So there are two players in metabolisms, and these are enzymes and adenosine triphosphate or what we called ATP. So we need to identify the role of ATP. So the functions of enzymes is to carry out critical tasks. This involves muscle growth, removing toxic and tearing down the molecules in food throughout digestion. So uh, enzymes are protein that help speed up metabolism or the chemical reaction in our body. So they build some substances and break other down. All living things have enzymes. Our body naturally produce enzymes, but enzymes are also in manufactured product and food. So how do enzymes function? So that is what I said. Enzymes perform critical tasks of lowering a reaction activation energy. That is the amount of energy that we must put in a reaction to begin enzyme work by binding to reaction molecules and holding them in such way that chemical bond breaking and bond forming process take place more readily. So enzymes create reaction in body. They actually speed up the rate of the chemical reaction to help support life. So the enzymes in your body help to perform very important tasks. So the second is the adenosine triphosphate or the ATP. So what it is? Let me define what is ATP. It is a source of energy for, for use and storage at the cellular level. The structure of ATP is a nucleoside triphosphate consisting of a nitrogenous base, adenine, a ribose sugar, and three cellular bounded phosphate group. So, adenosine triphosphate, also known as ATP, is a molecule that carries energy within cells. It is made energy currency of the cells. So it is in the end product of process of photophosphorylation and cellular respiration and fermentation. So um what is why ATP is important? ATP plays a critical role in the transport of micromolecules such as protein and lipid into in and out of cells. So the hydrolysis of ATP provides the required energy or active transport mechanism to carry such molecules across concentration and height. So the so shall we move on to the second unit? So it is the unit 6. So the unit 6 entitled microbial growth. So this unit is so difficult for me, but I need to explain. So, microbial growth refers to an increase in number of cells rather than an increase in cells size. Many microbes are unicellular, meaning they are made up of only one cells. So, what causes of microbial growth? One of the causes of microbial growth is warmth, moisture, pH level, and oxygen levels are four big physical and chemical factors affecting the microbial growth. So, in most building, warmth and moisture are the biggest overall issues present. So, the microbial growth on a solid substrate in which the substrate itself acts as a carbon source of defined as solid substance fermentation. So while SSF is the fermentation process occurring in the absence of near absence of free water among the particles using natural substrates. So and the last one unit is the unit 7. The unit 7 entitled Microbial Control. So this unit is so interesting. So define the following keywords related to the microbial control. Later I will give some keywords or terms. So but first let me define microbial control. Microbial Control of microbial growths means to inhibit or prevent growth of microorganisms. This control is effected into two basic by killing microorganisms and by inhibiting the growth of microorganisms. So what is the best way to control microbial growth? Uh, the best way to control the microbial growth is the most heat. Kills microorganisms by uh, coagulating their proteins. In general, moist heat is much more than effective than dry heat. Boiling heat to 100 degrees Celsius or more at sea level kills vegetative forms of bacteria, pathogens, almost all viruses and fungi and their spores with 10 minutes or less. So these are the key terms of related to microbial control. First is sterilization. It is a term referring to any process that eliminates, remove, or kill all forms of microbial life, including transmissive agents such as fungi, bacteria, viruses, and spore forms present of a surface contained in a field in medication rather or in a compound. So the process of disinfection inactive most microbes on the surface of vomit by using antimicrobial chemicals or heat because some microbes remain. The disinfected itself is not considered sterile. Ideally, disinfectant should be fast acting, stable, easy to prepare, inexpensive, and easy to use. So, what is the most effective method of microbial control? Heating is a widely used and highly effective method for controlling microbial growth. Dry heat sterilization protocols are used commonly in aseptic techniques in the laboratory. However, moist heat sterilize is typically a more effective protocol because it penetrates cell between than dry heats. So, what are the four factors that affect the control of microorganisms? First, warmth. Second, moisture. Third, pH level. And last is the oxygen. That's all. Thank you.